Histology of the salivary gland. So salivary gland is covered by a capsule. From the capsule, it sends a septa within the gland, and it divides the gland into lobes and lobules. So within the lobules, there is a connective tissue septa. So there is a secreting part of the glands, that is the acini. So two type of acini is there, serous acini and mucous acini. When we compare the serous and mucous acini, so the mucous acini, it is a larger, rounded in shape, and it is a light in stain. And mucous acini, the cell within the acini, it is a small in number compared to the serous acini. In lumen, it is a larger. Now the within the cell nucleus, the nucleus is present towards the basement membrane, and appearance of the nucleus, it is a flat, and the acini is stained lightly. So it appear as a empty appearance or vacuolated appearance. So nucleus is flat and it lies towards the basement membrane. So we are able to see the margin of the cells within the acini. So this one is the mucous acini compared to the serous acini. Now the serous acini, it is a smaller in shape and it is a darkly stained, and nucleus is rounded and it placed into the basal part of the cell. So within the cell, the nucleus is rounded and it towards the basal parts. Now, there is a large number of cells within the serous acini, and lumen is obliterated and the cell is darkly stained. And in the microscope, we are not able to see the margins of the cells, so we can able to see the rounded shaped nucleus, and it is a darkly stained. So septa divides the gland into lobes and lobules. So within the lobe, there is a intercalated and striated duct is present. So it is lined by the cuboidal to columnar epithelium. Now the in the connective tissue septa, between the two lobes, there is a interlobular duct is present that lines by the columnar epithelium, and when it passes towards the main excretory duct, that is a lobar duct and main excretory duct. So from that, it is lined by the columnar epithelium to the stratified columnar epithelium. Here it is a fat cells. Within the acini and within the connective tissue septa, there is a blood vessels lies. So blood vessels travels through the connective tissue septa. So here we are drawing the rounded shaped nucleus within the acini and towards the basal part. So this is the serous acini. Apical portion of the cell that contain the secretory granules, that is a zymogen granules in the serous acini. Three type of gland present in our body. The salivary gland, it is a serous gland, mucous gland and mixed gland. So parotid gland, it is a example of the serous gland. So pure serous acini are present in the parotid gland. So here we are drawing the slide of the parotid gland, slide of the serous gland. Now the submandibular gland. Submandibular gland, it is a mixed gland. So both serous and mucous acini is present, but predominantly serous acini are present in the submandibular gland. And sublingual gland. It is also a mixed gland, but predominantly mucus acini is present. Serous demilunes that presents as a cape over the mucus cell. It is known as the serous demilunes. So it is serous demilunes. It is formed by the swallowing of the mucus cell that pushes the serous cell peripherally, and there is a crescentic shaped serous cell is present over the mucus cell. Now here we are drawing the interlobular glands that is lined by the columnar to stratified columnar epithelium, blood vessels that lined by the squamous epithelium, simple squamous epithelium. The duct within the lobules, there is an intralobular duct that is lined by the cuboidal epithelium. So here we are drawing the mixed gland. So here we are drawing the predominantly mucus cell. So the predominantly mucus cell is present in the sublingual gland. Rounded nucleus that present into the serous acini, and it is a darkly stained. Mucus acini, there is a small number of cell is present. There is a flat nucleus towards the basement membrane and large lumen compared to the serous acini. In salivary gland, there is a presence of the parenchyma and stroma. The parenchyma consists of the secreting part, that is the acini, and conducting part, it is a duct. So the smaller duct united and it forms the larger duct, and terminally there is a excretory duct that excrete the product into the oral cavity that help into the absorption of the digested food. Excreted material from the acini, it is carried by the duct. The duct system, it consists of the intercalated duct, striated duct, intralobular duct, interlobular duct, interlobar duct, lobar duct, and main excretory duct. And stroma. The stroma, it consists of the capsule, connective tissue capsule that covers the gland, and connective tissue septa that presents within the gland, that divides the gland into lobes. So the between the acini and duct system, the connective tissue is present, and blood vessels travel through the connective tissue septa. Labeling: serous gland. The parotid gland, it is a pure serous gland. Sublingual gland, it is a mixed gland that contains the predominantly mucus acini. Mucus acini that present in the sublingual gland. Serous acini that present in the both gland. Serous demilunes. The serous cell cape over the mucus cell. It is a crescentic shape in intralobular duct within the lobules, interlobular duct between the two lobes, blood vessels, connective tissue. So between the two lobes, it is an interlobular connective tissue. Fat cells, it is a adipocyte. And in submandibular gland, it is a mixed gland but predominantly mucus acini is present.